Hello and welcome to ResearchPod. Thank you for listening and joining us today. Myalgic encephalomyelitis, also referred to as chronic fatigue syndrome or MEcfs, is a complex, long-term and highly debilitating condition. The causes and associated mechanisms of the condition have been poorly understood until now. Professor Sonia Marshall-Grodisnik, Director at Griffith University's National Centre for Neuroimmunology and Emerging Diseases, sheds new light on the pathological processes underlying MEcfs, paving the way for future pharmacotherapeutic interventions. In previous years, MEcfs was commonly referred to as an illness hallmarked by debilitating fatigue that is not alleviated with rest. However, overwhelming chronic fatigue is just one symptom of many. A defining factor of MEcfs is known as post-exertional neuroimmune exhaustion, which is the devastating exacerbation of multisystemic symptoms experienced by individuals with MEcfs. These symptoms vary in severity between patients and can include impaired memory and concentration, widespread pain, neurosensory disturbances, orthostatic or circulatory intolerances, sleep disturbances, immune dysfunction, gastrointestinal upsets, and temperature intolerances, all of which significantly affect patients' quality of life. MEcfs is thought to affect up to 24 million people worldwide, but the causes and pathology of the condition are difficult to pinpoint. There is still no routine diagnostic test, and diagnosis is based on the fulfillment of symptom criteria. Some symptoms can be treated or managed, providing relief for some patients. However, an effective treatment is lacking, and many MEcfs patients report intolerance to many medications. Aiming to change the landscape for MEcfs is a team of researchers at Griffith University's National Centre for Neuroimmunology and Emerging Diseases, NCNED, led by Professor Sonia Marshall-Grodisnik. Through their research, the NCNED team have revealed a wealth of new knowledge about the pathophysiology behind the condition and the disordered physiological processes associated with MEcfs. Their breakthrough findings provide hope of developing therapeutic interventions to treat and alleviate symptoms of MEcfs. Early groundbreaking research in the NCNED laboratory linked exposure to trauma or infection with the development of MEcfs. For many years, researchers have debated underlying mechanisms of the syndrome, leading some to speculate whether it has a psychological basis. This important study confirmed earlier research that MEcfs is a biological condition and not a psychological one. A significant finding from the NCNED research team was the fact that MEcfs is caused in part by the dysfunction of the transient receptor potential, or TRP family of ion channels. Also known as the TRP threat receptors, these channels come to the fore when the body is under any kind of threat, such as infection or trauma. This superfamily of ion channels garnered international recognition in 2021, when the Nobel Prize in Physiology or Medicine was awarded to two researchers who discovered that these molecular transducers play an important physiological function. Found mostly on the plasma membrane or cell wall, they are involved in the perception of heat, cold, and mechanical stimulation, and have enhanced our understanding of how the nervous system codes sensory information. Dysfunctions of these ion channels, which allow the passage of ions across individual cells, are known as channelopathies and are implicated in many diseases. In a series of elegant studies, the NCNED team confirmed MEcfs as a class of metabolic disorders known as TRP channelopathies. The team investigated the role of a specific transient receptor potential ion channel in immune cells in MEcfs. TRP ion channels have a vital role in calcium signaling in and out of cells, including in natural killer, NK cells. NK cells are a type of white blood cell that play a key role in the immune system. to kill tumour cells or cells infected with viruses, target cells. Specifically, the NCNED researchers demonstrated that the calcium ion channel, transient receptor potential melastatin 3, TRPM3, is impaired in MEcfs. As Marshall Grodisnik describes, the science behind TRPM ion channels is complicated. These are a group of 29 threat receptors comprising of 7 subfamilies. TRPM3 is one of at least 8 variants of the TRPM ion channel and likely has approximately 12 variants or isoforms. Either genetic mutations found in the TRPM3 gene, coding for the TRPM3 ion channel, or changes to the TRPM3 function through acquired factors such as trauma or viral infection are implicated in the development of MEcfs. Present in various organs and tissues, including the pancreas, brain, kidney, and eyes, TRPM3 ion channels are a part of pain transmission and heat-sensing pathways. Therefore, any dysfunction in such ion channels has widespread multi-system effects, which helps to explain the variety of symptoms presented in MEcfs patients. Marshall Grodisnik explains, MEcfs patients have experienced marginalization and disbelief with their illness in the face of profound disability and loss of activities of daily living. Validation of TRPM3 as the fundamental pathophysiology of MEcfs paves the way for easier access to diagnostic tests as well as to a potential array of pharmacotherapeutic interventions. The NCNED research team carried out delicate whole-cell patch clamp experiments to explore TRPM3 function in MEcfs patients. Placing a tiny micropipette onto individual cells provided accurate measurements of the electrical properties and function of these cells. These experiments demonstrated that TRPM3 channel activity is impaired in MEcfs patients. This channel activity impairment in NK cells affects the influx of calcium into the cell. That is, it disrupts its calcium regulation. Apoptosis, or programmed cell death, is dependent on calcium regulation, so the ability of NK cells to cause programmed cell death is reduced in patients who have dysfunctional TRPM3 calcium ion channels. MEcfs patients report benefit and relief of symptoms using naltrexone, but until recently there have been no studies demonstrating how naltrexone alters the pathophysiological mechanisms of MEcfs, the abnormal functional changes in the body that accompany chronic fatigue syndrome. In 2019, Marshall Grodisnik and colleagues published the first study confirming the potential utility of naltrexone hydrochloride as a treatment for MEcfs and described the mechanisms of action. Promisingly, their research demonstrated that incubating NK cells from MEcfs patients with naltrexone hydrochloride restored the function of TRPM3 ion channels when the NK cells were stimulated with interleukin-2, IL-2. IL-2 is a cytokine important in white blood cell signaling in the immune system. It aids NK cell interaction with target cells and improves cytotoxic abilities. More recently, further NCNED studies confirm the efficacy of naltrexone hydrochloride by demonstrating how it alters the pathophysiological mechanisms of ME. Opioid receptors, present throughout the body, are activated to regulate pain and inflammatory pathways. However, research shows that they also inhibit the function of TRPM3 and thus alter calcium concentrations, as well as affect NK cell functioning. Naltrexone hydrochloride is an antagonist of the opioid receptor and stops it from inhibiting TRPM3, therefore restoring TRPM3 activity, calcium concentrations, and normal NK cell function. By clearly demonstrating the mechanism of action of naltrexone hydrochloride on restoration of TRPM3 activity in laboratory experiments, their study serves as a precursor and basis for the design of future clinical trials into the therapeutic benefits of naltrexone hydrochloride for MEcfs. The NCNED researchers have also showed for the first time that stimulation of NK cells with IL-2 significantly improves their cytotoxic abilities in patients with MEcfs. Their studies reveal communication between TRPM3 signaling and IL-2 signaling pathways, which are important in immune regulation. Discovery of such crosstalk between these two pathways provides information into their functioning and further understanding of the pathophysiology of MEcfs. The team developed a new immunofluorescence method to determine where TRPM3 and PIP2 reside within the cell. PIP2 is a membrane phospholipid molecule known to regulate several physiological functions, including ion channel activity. Using one fluorescent dye to tag TRPM3 and another to tag PIP2 allowed the researchers to accurately visualize these two molecules under a microscope. Excitingly, they found that TRPM3 and PIP2 are located very close to one another in NK cells. Co-localization like this suggests a strong functional connection between the molecules. PIP2 is a membrane phospholipid molecule known to regulate several physiological functions, including ion channel activity. Their study showed changes in TRPM3 and PIP2 co-localization in MEcfs patients compared to healthy subjects, suggesting that impaired TRPM3 function in the condition is related to PIP2, which itself has a role in NK cytotoxicity. Importantly, the research further elucidates the intricacies of the impaired TRPM3 ion channel function pathology and implicates PIP2 and IL-2 in this mechanism. NCNED's research is forging the way ahead in improving our knowledge of the pathophysiology of MEcfs. Finally, patients and researchers have an insight into the causes of this debilitating disease, which have remained elusive for so long. Such studies not only provide crucial information into the pathophysiological mechanisms, but also provide a foundation on which further mechanistic research can be conducted. This vital research paves the way for diagnostic tests and enables pharmacotherapeutic interventions to be explored, offering much-needed relief for patients. Thanks for listening, and stay subscribed to ResearchPod for more of the latest science. See you again soon.